curiosity. And for those of you that are actually going a pre-health route, it does give you the opportunity to interact with another demographic, the elderly, that you potentially could see in the future in your own practices. So again, it's Florida Atlantic AdaptedAquatics.com. Click on the link, A Bear Because We Care, and go ahead and sign up and come help us make some teddy bears. Thank you, you guys. Thank you, Dr.
Sure, you're welcome. Okay, let's go ahead and get going because we've got a lot of ground to cover. So we're going to have the third exam on next Monday. It'll cover chapters 11 through 16. We're going to do 15 today. Hopefully, with a little bit of luck, I'll just start into 16. And then we'll get 16 on Wednesday. We'll be back on track, and we'll be in the home stretch at that point. If I'm looking at the academic calendar correctly and they haven't changed things, they have a way of changing things, the withdrawal deadline will be a week from Friday. So I will have grades posted up before that time. You'll now have, you can see your average of 3 exams. Keeping in mind that you do have that optional final exam, then you can make your decision. Just keep in mind that if you go past that withdrawal deadline, my ability to help you is diminished significantly. It's a petition process after that. The best I can do is sort of write a letter of support, but it's not necessarily a foregone conclusion if you go for a late withdrawal. Okay. So everybody by now, you know the drill. We'll just be ready for Monday. Also, the sample questions for exam 3 are up on Blackboard. I got a little carried away. I think I've given you nearly 25% of the questions from the exam. Plus, there's about 22 sample multiple choice questions.
Thank you.
Thank you. Well, let's see what you say after the exam. Okay. So we're doing chapter 15 today, and maybe we'll get a little bit of a start on chapter 16. So let's do the 2 questions that we've been typically doing now. So for those of you who are math literate, you realize that you pick up about 80% of all the iClicker points within the first 3 to 4 minutes of class. Assuming you stay for the rest of the questions too. It looks like we'll only need 1 minute for this one. Coming in pretty fast. And again, if you want the participation points, make sure you put something in, even if it's wrong. All right. A few more seconds. All right. What's the answer? I keep on it. Okay. Oh, let's just not tell them. All right. So this is what I was saying last time, that as you get into more developmentally complex organisms, you tend to have more introns in the genes, and those introns tend to also be much larger. Okay, so from today we'll be talking about protein structure, so let's try this question. We'll go for 1 minute again. Okay. And the answer is? Okay, secondary structure. So we'll explain all this in more detail in a minute, although you should have seen this probably from other courses. Okay, so we've looked at DNA replication. Then we went through transcription, and then we talked about the processing of the pre-mRNAs in eukaryotic cells, the capping of the mRNA, the poly-A tail, the splicing of the introns. We'll assume we didn't talk about the actual transport of the mRNA, so we'll assume that the mRNA has now been transported out of the nucleus into the cytoplasm, where it is to perform its major function, that is to interact with the ribosomes and the tRNA in a cooperative way that results in the synthesis of a polypeptide. So this is this process of translation, and fundamental to that process is an understanding of the genetic code, really getting to the heart of how genetic information is encoded ultimately in the DNA sequence. All right. So just a little bit of a history. We're not going to go through these experiments, but back probably in the 30s to 40s, somewhere around there at Indiana University, Beadle and Tatum did a classic set of experiments with Neurospora, which is a type of fungus. It was a major genetic model system for many, many years. It hasn't been used too much in like the last 50 years. But they had this important idea. They were looking at mutations in biochemical pathways. We've talked about this a little bit before, that you might have basically when we talk about complementation. So you might have 5 different enzymes that are needed to make compound X. Mutations in genes that encode any of those enzymes can block the pathway. And through these experiments, they came to the conclusion that each gene would encode a single enzyme. The reason I put this up here is to show how we've had to modify this idea over time. They didn't quite understand the structure of a lot of enzymes, that those enzymes are multimeric, that there's multiple subunits, and so it had to be modified that each gene codes for a single polypeptide. Then with the discovery of the introns and self-splicing, with differential splicing that we talked about last time, it became clear that now we have to say that each gene can code for 2 or more polypeptides. They're closely related, but they are distinct, and so we call these isoforms or variants. So it's just changes in our thinking about really what a gene encodes as we've learned more about the molecular basis of this genetic structure. You're already familiar with, so proteins are the workhorses in the cell. They do everything from enzymes, structural components, regulation, defense being part of the immune system, communication, cell surface receptors. So they are basically, we are a collection of about 200 different cell types. Those cell types really differ based on the particular proteins that are being expressed and active in those cells. So this whole process is fundamentally important. It all boils down to these 20 amino acids that are commonly used in proteins. Occasionally you find some modified amino acids in proteins, but extremely rare. So we'll just, for our purposes, we'll limit ourselves to the 20 common amino acids. They're summarized in figure 15.5. We're going to talk about them in just a minute, but I'm not going to ask you to memorize the structure of those amino acids. Again, I let the biochemists do that, and they probably do it very well. But I am going to say that this is the time to learn the 3-letter and especially the 1-letter codes for these amino acids. Now that so much of our information comes off of databases, either direct protein sequences or deduced protein sequences, and it's always provided to you as the 1 amino acid code. So in research papers, it's almost ubiquitous now that protein sequences are identified strictly by the 1-letter codes. Most of them are obvious, like W for tryptophan, but some of them are, a little more obscure, like M for methionine. But you'll see. You probably should already be familiar of this sort of central structure of amino acids. So the alpha carbon in the middle, in carbon forms, always has 4 bonds. So we have a hydrogen, an amino group, a carboxyl group. This part, these 3 bonds, those are the same on all the amino acids. They only differ at the business end, the so-called R radical group. Although given current politics, we might have to change that name too. We'll call it the side chain. Okay. What I want you to do is I'm going to show you the 20 amino acids grouped together based on the common biochemical properties of their side chains. I want you to think about them in terms of that kind of grouping, because later on we're going to talk about genetic mutations that change an amino acid. And these are the ones you already know about, the sense mutations and such. If you change an amino acid within the same group, will you probably affect the activity significantly? Let's say as opposed to changing it to another one and a much more radically different. If you have an amino acid that's negatively charged and you change it to another one that's negatively charged, you might have minimal impact on the functionality of that protein. But if you change it from a negative charge to a positive charge, you're more likely to have a difference. So I want you to think about these in groupings in the context of what kind of changes would probably have minimal effects. And I say probably because there will be exceptions. And which ones will undoubtedly have a major effect on the structure and function of the protein. I'll show you a couple of those amino acids that are particularly important. So these are the nonpolar aliphatic groups. All right, so are these going to be hydrophobic or hydrophilic? These are going to be hydrophobic, so they're going to be polar groups. Nonpolar, the aliphatic. And a lot of these amino acids tend to be on the outside of the proteins, because most proteins in general, the reactive side is sort of a pocket up inside the protein. And so interchanging glycine, alanine, valine, leucine, or isoleucine probably would have minimal impact on the functionality of the protein. Methionine is in this group, but I put a note in here that one, changing it to or away from a methionine, will likely have an impact. And that is, methionine is a fairly rare amino acid, and it's very often part of the active site or the substrate binding site or whatever. So methionine often has a key role in the functionality of the enzyme. So changing it to anything else or changing something else to methionine, I would predict it will have a significant effect on the functionality. So you got the nonpolar aliphatic group, then the ones with the aromatic ring structures. So you have phenylalanine, tyrosine, and tryptophan. Again, interchanging any of these probably would have minimal impact. The polar uncharged group. So would polar be hydrophobic or hydrophilic? They're hydrophilic, they're just not charged. Okay, a couple of key ones here that would be important. Cysteine adjacent cysteine, or within the three-dimensional structure, two cysteine amino acids can form a disulfide bond. This is critical in the formation in the three-dimensional structure for a lot of proteins. So especially changing away from one of these cysteines to something else would probably start to disrupt the structure of the protein, because the stabilizing disulfide bond can't form. So here I would put the cysteine kind of off in a different category, and changing it to or away from a cysteine probably would have some kind of an effect. The other one that will almost always cause a change is in proline. Proline is unique among the amino acids. If you notice, it actually doesn't have the free amine group. All of these have the free amine group, except here it's part of the ring structure. Generally, whenever there's a proline in the polypeptide, it causes a bend in the polypeptide. So changing a proline or inserting a proline or changing something else to a proline would probably have a significant effect on the protein. Then we have the positively charged groups. Lysine and arginine, you saw those where? They're prominent in the histone. All right. Histidine is a little tricky. It's normally positively charged, but the equilibrium for it is very, very close to the peak, So basically, the pKa is very close to pH 7, to the physiological pH 7. The take-home message is whether the histidine is positively charged or not can differ by very small changes in the intracellular pH. So in some cases, proteins use the histidine in a regulatory sense by manipulating the microenvironment of the cell and making small changes in the pH. So if that histidine is part of this regulatory mechanism, then changing that would have an effect on the protein. If it's not part of that, then it's hard to say. So it's just sort of a qualifying point to it. But again, I'm not going to ask you to draw the structures, but rather to be able to recognize the groupings of these so that if I change arginine to valine or I change lysine to histidine, will that or will that not probably have some impact on the polypeptide? Okay. Pretty straightforward. All right. Oh, that's right. And negatively charged one, I almost forgot those, aspartate and glutamate. Okay. So interesting thing, I said memorize, learn. Memorize means you'll forget it by 4 o'clock on Monday. The one-letter codes, interestingly, so will there be one of those questions on the exam? Yeah, write it down. There will be. Nearly everyone gets that right, regardless of their score on the whole exam. They do seem to be able to manage that. I'd be the one who'd get it wrong. Okay. So, amino acids, we're really talking about polypeptides. So just like with nucleic acids, we had RNA and DNA, these are polymers. They're repeats of a basic subunit. Instead of nucleotides, now we have amino acids. Instead of a instead of a phosphodiester bond, we have a peptide bond that joins these amino acids together. So they're just a linear polymer of amino acids. So that's really what the amino acid coding sequence in the mRNA is doing, is just specifying the sequence of amino acids, and each one is going to be joined by this peptide bond. Just like we had with DNA and RNA, there is a polarity to these polypeptides. There's a free amino group on one end, often called the N-terminus. All right, that'll be on the first amino acid to be the first amino acid that's in the polypeptide, and at the other end, the last amino acid just before the stop codon will be a free carboxyl group, or oftentimes just called the C-terminus. So a lot of times you hear people refer to the N-terminus and the C-terminus. It's the same analogy to the 5' and the 3' ends. There is a polarity to these proteins. The formation of this peptide bond, if I remember my chemistry right, is basically a dehydration reaction where this carbonyl group is going to interact with the amino. So this is the polypeptide chain. The new amino acid is coming in, and then there's a loss of water and formation of this peptide bond. And then that just gets repeated over and over again. Okay, so a lot of this should be familiar. All right. What we're really more interested in is what's the structure of these polypeptides and what's their functionality, and how does regulation of the expression of the genes that code for these polypeptides affect the biological outcome. So we're going to look at 4 structures: this primary, secondary, tertiary, and quaternary structure. Just like we had with DNA, the primary structure is simply the sequence of amino acids. But the sequence of amino acids, does that tell you what the 3-dimensional structure will be? Not directly, but some software, simulation software is getting really good now at being able to predict what from that primary sequence, what that protein will fold into. But by itself, the primary amino acid sequence is just that. Do we actually have to sequence these proteins to have that primary amino acid sequence? When you download an amino acid sequence for a protein from GenBank, is that because the protein was necessarily sequenced? Where can we get that information? Buy it? No, it's all free. All right, what are we more likely to So there's a lot more DNA data in the databases than there is protein data. And once we know especially once we know what the mRNA sequences are, then we can simply predict, based on the genetic code, what the amino acid sequence would be. So for a lot of proteins, it's a conceptualized amino acid sequence. But given the universal nature of the genetic code, it will be very accurate. Then we start to get into the three-dimensional structure. The secondary structure is really just two organizations of the amino acids referred to as beta-pleated sheets and the alpha helix. These are they are substructures, small substructures within the protein. They just occur so often. I'll show you in a few minutes how they're the interaction of hydrogen bonding, somewhat analogous to what we saw in double-stranded nature of DNA. But they are very, very frequent, and they make up subcomponents of what we're going to call motifs. I'm going to make this as complicated as I can. We're going to have motifs, which are also substructures of the final protein. Okay, we'll see if we can get there. When we get to the tertiary structure, we're talking about its overall three-dimensional structure. Okay. To do this accurately, you have to be able to crystallize the protein and then do X-ray diffraction and mass spec and a lot of detailed analyses on it. So there's an increasing number of these proteins are available, but it's still very, very small compared to the amount of DNA data that we have. If the enzyme functions as a single polypeptide, then we're done. DNA polymerase 1 is a single polypeptide, so it doesn't have a quaternary structure. The quaternary structure comes in these multimeric enzymes where you have multiple polypeptides that come together. Would DNA polymerase 3 have a quaternary structure? You can nod yes. Okay, it's okay. How about RNA polymerase? How about RNA polymerase from E. coli? Would it have a quaternary structure? It's about 6 subunits, right? So hemoglobin would have a quaternary structure because it's 4 globin polypeptides. So if you have 2 or more polypeptides that have to interact with each other to form the final functional enzyme, that's what we mean by the quaternary structure. If you function as an enzyme and you're just a single polypeptide, then you stop at tertiary structure. Any questions? Okay. So I found these on Dr. Kaiser's microbiology website. I thought they would show things pretty clearly. These are the 2 secondary structures. So you'll see them coming up over and over again as we look at more detail of the three-dimensional structure of the proteins. They are, in fact, the result of repeating structures through hydrogen bonding. So that's what it's showing you in a manner it's not the same, but analogous to what's holding the double-stranded DNA structure together is you have these hydrogen bonds between either an oxygen or a nitrogen and a hydrogen. So you have these so-called beta-pleated sheets and the alpha helix. So that's the secondary structure. To try to show you this in a little more kind of in a progressive sense, we start out with the primary amino acid sequence. Then the secondary structure, we have an alpha helix and just this portion of the protein. Here we look at the tertiary structure, which is just one of the polypeptides in 3-dimensional space. And since this looks like a heme porphyrin system, I'm going to guess this is globin. And then the final structure when all 4 of those polypeptides come together to give you the final mature functional enzyme. Okay? Again, if the enzyme or the protein, whatever it is, whether it's a structural protein, an enzyme, whatever, if it's a single polypeptide, you do not have a quaternary structure. That is only when you have a multimeric enzyme that has 2 or more polypeptides that have to interact with each other. All right. Any questions on just that general? Again, that I would expect a lot of this to be familiar from other classes. Just sort of getting everybody up to speed. Yes. That's what I'm guessing. I'm going to make a guess. This is some, additional factor. I'm guessing it might be, for instance, the heme porphyrin ring system and globin. So a lot of proteins have to have additional metals and such associated with them to make them functional. In this figure, it's probably distracting. So this is not important to the discussion. You just have the tertiary structure. An important point, maybe not an important point, I don't know. If you have a multimeric enzyme, are they always different polypeptides? No. So, for instance, beta-galactosidase, which we'll talk about, probably not till next time. Okay, it's the same polypeptide, but it takes 4 copies of it to form the functional enzyme. So a multimeric can be multiple copies, and there's lots and lots of examples, multiple copies of the same polypeptide. But as long as there's 2 or more in the final enzyme, it'll have a quaternary structure. Okay, the genetic code. So you should already be aware that it was worked out that you have the so-called codons. So this is a sequence of 3 nucleotides. Is the codon in the DNA or the RNA? The codon is technically in the RNA. A lot of times we will talk about, and I may slip into this, I'll say ATG as a start codon. That's incorrect. It comes from working with DNA sequences and converting them into RNA. The start codon would be AUG. So these codons are RNA sequences. The genetic code was cracked in the 1960s by Marshall Nierenberg at the NIH. This was one of the major breakthroughs in the early days of molecular genetics. Actually, Dr. Weisberg, who I've mentioned several times and still collaborate with, he's still working up in Jupiter. He was just down the hall from Marshall Nierenberg's lab and then collaborated with him on another project at the time when Nierenberg did this work. But it was really good, nice set of experiments. We just won't go into them right now. So basically, the genetic code was cracked in the early 1960s. So the first thing was, what's sort of the minimum number of nucleotides it's going to have to code for these amino acids? If you have 20 amino acids, could you code, and you have 4 nucleotides, what would be the minimum number of nucleotides that would have to make up a codon? It would have to be 3. With 2, what would happen if there was only 2 bases? How many could you do if you only had 2 bases? 4 to the second or 16. So it had to be at least 3. So there's 4 to the third or 64 possible codons. Would the cells have to actually use all of them? Not necessarily, but they actually do, to an extent. So there are 61 codons that actually code for amino acids. So in one form or another, all 61 of those are used. The other 3 are going to be used for the stop codons. So these are all the so-called sense codons. A sense codon just simply means it specifies an amino acid. The code is said to be degenerate, and you can have your own implications for that. In the scientific sense, what it means simply is that the amino acids can be coded for by 2 or more codons. So we have 61 possible codons, but we only have to code for 20. So we can use multiple codons for the same amino acid. And that's true for most cases. Tryptophan and methionine are the only 2 amino acids that just have 1 codon each. All right. So, if instead of 20 amino acids there were 200 amino acids, what would be the minimum number of bases in a codon? Since this involves math, I'll give you a minute and 15 seconds. Okay. So, what would be the minimum? You're not as math challenged as you thought. Okay. So any questions there? So you'd have, if you had 4 codons, you'd have 256 possibilities. Okay. All right. All right, stay with me here because a little bit this will get a little bit confusing. So some of the amino acids have as many as 6 codons. The majority have 4. They're called synonymous codons. It just simply means this is the group of codons. They all code for the same amino acid. Now the challenge is the tRNA has to have an anti-codon loop that's complementary to the codon. So if we have 6 different codons for the same amino acid, we're going to have 6 different tRNAs that have the same amino acid, but each have a different anti-codon. With me on that one? Say that again. If we have 6 possible codons for a specific amino acid, we're going to have 6 different tRNAs that all have the same amino acid, but will have different anticodons because that is what base pairs with the codon on the mRNA. So these are called isoaccepting tRNAs. They are different tRNAs. They're each coded by different genes, but they all carry the same amino acid. All right. The other thing that's come out of a lot of molecular studies is there's 61, we said there's 61 amino acid coding codons, and there are many examples of organisms where we know that all 61 codons are in one way or another used. But for any given organism, any particular species, a lot of times what you find is they actually only use 30 to 50 of those possible 61 tRNAs. So most cells make fewer tRNAs than you would think would be necessary with 61 different codons. So how do you get by with no more than 50 tRNAs when you may have 61 possible codons? And the answer is you get a little bit sloppy. So this is the idea of the wobble. So if you look at the synonymous codons, everybody's memorized the genetic code, right? That table. Everybody's got that memory. You do have to have that. You will know it by Monday, right? Okay. Now, if you want, I'm not suggesting anything, but your colleagues who aren't here today, if you told them, I don't know. Back long before we had calculators and I was in high school, you guys probably don't even know this, but you know how to do, everybody knows how to do logarithms, right? You punch a number on your calculator and you get some number comes out of it, right? Well, back in the dark ages, before we had calculators that actually calculate the log, there were log tables that you had to look up what the logarithm was for your particular number. And I got this close to convincing one guy in the math analysis class that you're going to have to memorize those log tables, that there was no way the teacher was just going to give you the log tables. I almost did it. Okay, so here's what happens. If you look at the majority of amino acids have 4 codons. There's 4 synonymous codons. They differ. Let's put it this way. When you have synonymous codons, most of the time the first and the second base are different. Are the same. The third base is different. So you'll have 4 codons that code for amino acid X. The first 2 bases will be the same, but the third could be A, U, G, or C. What happens here is that pairing at that third position is a little bit sloppy, which allows base pairing, unconventional base pairing. So the same tRNA can code for more than one, can base pair with more than one codon. So this is called the WOBBLE effect. So here would be, so for instance, here's a serine tRNA. Here's the codon UCC. And it normally would base pair AGG. This would be the normal correct pairing. But over here in the third position, that same tRNA can also base pair stably enough to function in translation with UCU. So the same tRNA can read 2 different codons. By doing that, you don't need, the cell doesn't need to make all 61 tRNAs. Okay. Any questions? All right. Yes?
Would adenine or adenine be like a better, like instead of like it's using C to pair with G, like would A be a little bit better because it's more similar in structure?
Actually, it doesn't really seem to matter what this base pairing is. It's actually more in the biophysical structure of the way the tRNA is interacting with the RNA that it's not as precise in its base pairing requirements in that third position. Okay, so let's try this question. So what is the first nucleotide at the 5' end of the anticodon? All right, I'll go to 1 minute on this one. All right. What's the answer? Always anti-parallel. So the codon is the mRNA sequence. The mRNA is going 5' to 3'. This makes this the 3' base. Is that the 3' base in the codon is the wobble position. That would be the first base in the anticodon, because this is going 5' to 3' right to left. So always, these pairings are always, even with tRNAs, always anti-parallel. Okay, any questions? All right. The other thing that came out of cracking the genetic code and decoding a lot of these mRNA sequences is that there is no punctuation in reading the code. There are no spaces. It's read 3 nucleotides at a time. And it also means that any given mRNA has 3 possible reading frames. So the reading frame refers to really the start point. What nucleotide are we going to use to begin reading the first 3 nucleotides that will specify the first amino acid? So we can take, this actually is the same sequence. I've just changed the alignment and just using G here as a reference point. You have the same sequence, but in this case it's the first base of a codon. Here's the last base of a codon, and here's the middle base. So now you just read 3 at a time as you go along, and you'll get a completely different amino acid sequence. So this is really what happens when you have a frame shift mutation. You're inserting a base or deleting a base, and you change the reading frame. So the reading frame is going to be defined by the very first codon that the ribosome reads, the first 3 nucleotides, and the first amino acid that it specifies. Okay? All right, so, okay. So let's see if you can remember what I said 30 seconds ago. Oh, look, you're getting it right. Few lost souls, but yeah, 1 minute will be good enough for this one. Okay. So this is obviously what? Okay. All right. That was easy. This one, okay. Here's my prediction. Show me you're different than other classes. Majority of you are going to get this wrong. And you're going to want to do over. But go for it. And then we'll see if we can learn from it. Read it carefully. Well, 15% have it right. All right. Here, I'll bring this to a merciful end at 1 minute. Everybody got something in? Okay. Okay. 19% got it right. Should I show you or you want to do it again? You know, just do it, right? Yeah. 6. When you have, this is one of those you got to bring several ideas together. When you have the DNA sequence, you got 2 strands. We already talked about you don't know which strand codes for the gene, and then each strand would have 3 possible reading frames, so therefore there's 6. Now let's do it again. I don't think I could say that again. No. When you have just the genomic DNA sequence, you have 2 strands. As we talked about, the gene could be coded for on either strand. You don't know which strand is transcribed. So until you know which strand is transcribed, you've got 2 possible transcripts, and each transcript has 3 possible reading frames. So when you first have just the genomic DNA and you don't know anything about the gene, as here where it just has a segment of genomic DNA that contains that locus. You actually have 6 possible reading frames until you can get it narrowed down and know which strand is transcribed. Okay. If it makes you feel any better, the last 10 classes didn't do any better either. It's a teaching moment. Okay. Hopefully it'll help you on the exam, and it'll be worth a whole lot more than this 2 points or whatever it is. Okay. So getting into the actual process of translation, the key factor is the so-called start codon. So interestingly, and again, I think it argues as one of those very early evolutionary events, both bacteria and eukaryotes all use the same start codon. So the start codon, with a few exceptions being found now, but for our purposes, all bacterial and eukaryotic genes start with an AUG start codon. That's the one codon for methionine. So all prokaryotic and all eukaryotic proteins start with methionine. Bacteria have to do it their own way, so they use a modified methionine, a formulated, we're not going to go into the structure of it, but it's N-formal methionine. The important point here is, in bacteria, you only have N-formal methionine at that first position. If there's a methionine that's internal in the polypeptide somewhere else, and there's another AUG start codon, it will be regular unmodified methionine. In fact, there's a specific tRNA that's sole function is to be the initiator tRNA. So in bacteria, you have N-formal methionine. Now, the other thing is, a lot of times when you isolate the proteins, if you sequence those proteins, the methionine won't be there. In many cases, the methionine in both bacteria and eukaryotic cells ends up being removed in post-translation or after-the-fact modifications. We'll talk about that a little bit more in a few minutes. All right. In eukaryotic cells, they don't have N-formal methionine. They use the standard unmodified methionine at both the start codon and at any internal AUG codons in the polypeptide. Okay. Here's another one that a lot of you are going to get wrong on the exam. So I'm trying to help you out here. We have 3 termination codons. Those are the stop signals. When you hear about nonsense mutations, these are mutations that change a codon that code one of the 61 codons that codes for an amino acid to one of these 3 codons, which is the signal for termination. So these are called stop codons, nonsense codons, termination codons. They all mean the same thing. The key here is there are no tRNAs that pair with these codons. There are release factors. And again, you're seeing factors. It's going to be a protein. When you see element, it's going to be a DNA sequence. So there are proteins that interact with those stop codons for the purposes of ending translation and dissociating the ribosomes and releasing the mRNA. We'll talk about that process in more detail in a few minutes. There are no tRNAs that base pair with the anticodons. Okay. What came out of cracking the genetic code was the, at the time, thought to be fully universal nature of the genetic code. And so prokaryotes and eukaryotes, so everywhere from E. coli all the way to humans, uses the same genetic code, the same codons to code for the amino acids, except there are a few exceptions. They usually involve some stop codons. Most of those are in genes like in the mitochondrial genome and such. So just be aware there are a few minor exceptions to this universal nature of the genetic code. And it goes like everything else in biology. There's always the exceptions. But 99.99% of the genes use the same genetic code. Okay. This one may take a few minutes. Okay. So, by poly U, I mean that you're looking at just a repeating sequence that is just a series of U's. Poly C would just be a series of C's. So that's going to be your template. Or the random copolymer just means you have C and U put together in all different possible combinations. Does this look like it should probably be an extra credit question? Okay. I know you do 2 points. I'll give 5 points for this one. I don't care.
You don't care at
100 points? It's all relative, so it's not going to make any difference. Okay. Yeah, it looks like probably about a minute and a half ought to get all the answers in. Yeah, good. Majority are getting it right. Okay. So what's the answer? Good. No, I'm impressed. About 70, almost 75% of the class. And nobody answered E. Okay. So is there any questions? Anybody understand the question? No, no questions, or no, you don't understand the question? A and not. There are 3 different codons. Could you really conclude that?
I wasn't sure. I don't think so. No.
No. So A is really the only. So the difference is, and again, it comes to answering questions.
Okay.
B is true, but it's not the answer to the question. You know, it's the interpretation. What's the most reasonable interpretation that's completely consistent with what you know?
Okay.
Was there a question? Yes.
Based on this question specifically, couldn't C also be true? Because if poly U only contains U and poly C only contains C, that means the only codon is going to be CCC. So technically, for phenyl, are we not to codon, or there would only be one codon for each of those, it would either be U or CCC.
No. Okay. That's a good point. So the question. But why wouldn't that be true?
Because they probably come up with different codons. But
Yes.
I'm just thinking specifically for this question.
But to say that no, but that's a good point. You come up with this answer, then you conclude that there's only one codon for phenylalanine and proline, but you didn't put all possible combinations in there. So it tells you that CCC is one of the codons, but it doesn't tell you it's the only one. Okay, it's that sort of overextending for what you know. When this says there's only one codon, that would be your conclusion. But it could be, for instance, maybe 3 A's code for proline. You just don't have that data. Okay. Any other questions? Okay. So we're going to go through this process of translation. Again, you've probably seen it in general biology. At least you've heard some of the words. So like with some of these other things, I hope you have a better understanding of it once we get done with this discussion. Okay. So the process of translation is really the synthesis of the polypeptides. So it involves the ribosomes, and they're going to attach near the 5' end of the mRNA. And just like as synthesis was in the 5' to 3' direction, the ribosomes translocate or move along the mRNA in a 5' to 3' direction. We're going to break this down into 3+1. In terms of steps, we'll have the initiation, elongation, and termination, as we talked about with DNA replication and transcription. But we also have to add in tRNA charging because that's an integral part of this entire process. Okay. So stay with me here because, again, there's a little point here that's going to tend to trip you up a little bit. So the enzyme, so what we have to do is we have to make charged tRNA. There's amino acids sort of in a pool out here in the cytoplasm. There's tRNAs that have been recently synthesized, and they've been transported out of the nucleus and they're in the cytoplasm. We have to charge the tRNA. What we mean by that is we need to add the amino acid on the 3' end of the appropriate tRNA. The enzymes that catalyze that reaction are the aminoacyl tRNA synthetases. So they synthesize aminoacyl tRNA. That just simply means tRNA with its amino acid joined to it. Here's an important point. There are 61 different tRNAs. Cells may or may not use all of those. There's 20 different amino acids. There are only 20 different aminoacyl tRNA synthetases. The same amino acid, the same enzyme charges that amino acid on all of the isoaccepting tRNAs. So if you have 6 codons for a particular amino acid, that's 6 different isoaccepting tRNAs, but they all have the same amino acid. There's one synthetase that charges that amino acid on all 6 tRNAs. So each synthetase is amino acid specific, and it works with all of the isoaccepting tRNAs for that amino acid. Okay. So a few more details about it. The amino acid is always charged onto the 3' end of the tRNA. So all of the tRNAs end in this CCA sequence. And right here, and so here's that, here's that A, here's the phosphate and the phosphodiester bond. And right here on the 3' hydroxyl, where the next nucleotide would have been added, is where, through this carbonyl group, this amino acid is charged on. All right, so it's on the 3' end. Down here is the anticodon. And this is an energy requiring reaction, so it requires ATP, hydrolysis. Okay. So let's see if you can remember all that. Oh, 1 minute should be good. Okay. And the answer is, okay. But I stood up here and did everything but jump up and down. That's what my genetics professor, I remember his name was Dr. King, not the Dr. King. But that's what he said. If there was something really important, he would jump up and down on the desk. I'm not doing that, but that was his way of getting our attention. There's also one. I must have told you why. He was the one who influenced me to go into genetics. Didn't I say that story? Yeah, I thought I did. It gets old. I have a good friend of mine. He's older than I am, so we can tell each other jokes and everything else. And we can't remember whether we heard it or not from the day before. Okay. Here we get into a little bit of the details. So we charge the tRNAs. The first step is the initiation step. Just like with transcription, initiation is the rate limiting step in protein synthesis. So the efficiency with which the ribosomes interact with the messenger RNA controls how much protein is being made in per unit time for that particular messenger RNA. The more efficiently the initiation complex comes together, the faster elongation starts, the more polypeptides made. Okay. So in a bacterial system, we talked about this 5' UTR, the untranslated upstream region. And we said there was a sequence called the Shinan-Dalgarno sequence. This is critically important in bacteria because this helps the small ribosomal subunit interact with the mRNA. So in both eukaryotic cells and prokaryotes, you're going to have the small ribosomal subunit interacting with the mRNA first, and the final step will be the large subunit. So the whole the two subunits of the ribosome have to be dissociated to start this process. Okay? So we have some factors. IF just simply means initiation factor. So IF3 is actually here. It binds, if you notice, it binds to the small subunit right over here. Its real function is to keep the large subunit from interacting until this whole complex is finished, and the large ribosomal subunit is the last step. So you have the small ribosomal subunit interacts with the Shinan-Dalgarno sequence, placing it over the AUG start codon. Then we have to bring in the initiator tRNA. So since this is bacteria, this is going to be this formulated or F-met tRNA. There's a few more of these factors involved. So there's initiation factor 2 and GTP, not ATP. So there's a lot of these kind of processes in cells that utilize GTP rather than ATP. Not so much as an energy source, but as a part of the regulatory mechanism. So you're going to have GTP, initiation factor 2, and initiation factor 1 are going to also, and the tRNA are going to come in. So we end up with initiation factor 1 and then 3 on the small subunit. Initiation factor 2 and GTP are associated with the F-met tRNA, and it's hydrogen bonded to the start codon. Okay. Then all the initiation factors dissociate. That's triggered by hydrolysis of GTP to GDP. So only one phosphate is cleaved off, not the high energy phosphoanhydride bond, but that bond between the beta and the gamma phosphates. So this is more of a regulatory mechanism. And that then facilitates the large ribosomal subunit coming in, and now you've got initiation. The ribosome is associated on the mRNA. The F-met tRNA is in the right position, lined up with the start codon, and now you can actually begin elongation. Right. How this is how it happens in bacteria. It's very similar in eukaryotes with a couple of differences. Okay. First of all, eukaryotes don't have the Shinan-Dalgarno sequence. That's just in bacteria. But what they have is several factors, again, more proteins, that are going to actually interact with the cap structure. So we talked about, when we talked about the processing of mRNA, I made the point that that cap on the 5' end is required for translation. And so basically the ribosome is going to interact near the 5' end of the messenger RNA, and then it's going to scan along the mRNA until it finds the start codon. To help it find that start codon is a consensus sequence surrounding it called the Kozak sequence, named for Marilyn Kozak, who worked all of this out a number of years ago. And so this is going to tell the small ribosomal subunit the proper location on the mRNA. So it's really the cap structure, these additional factors that interact with the cap structure, and this Kozak sequence are doing what the Shinan-Dalgarno sequence is doing in bacteria. And as always, there's a lot more factors involved. All right. And more recently, but I mean, it's been a number of years now, initially this poly-A tail was thought to be just required for stabilizing the messenger RNA from degradation, but it actually also interacts with these factors, these cap-binding proteins, to help initiate this whole process. Now, going back into bacterial cells where it's simpler to explain, now we have to do elongation, which is just going to be the sequential addition of each amino acid as the ribosome is reading the codons 3 nucleotides at a time. So there's going to be the A site or the amino acyl site, the peptidyl site or the P site, or the exit site or E. And just to make it more complicated, there's 2 important elongation factors. So now it's EF instead of IF. And they're TU and TS, and also we'll have GTP. Okay. So here we go through this in a stepwise manner. So here's the initiation complex. It starts out with the F-met tRNA at the P or peptidyl site. The new tRNA, which is associated with elongation factor TU and GTP, is going to enter the complex through the acceptor or amino acyl site. You can call it the acceptor site too. It comes in at the A site. So here's our first amino acid. This one is now glycine. So it enters, it's here. All right. Then just like we saw before, the GTP is hydrolyzed to GDP, and EFTU is released. And then there's a peptide bond that forms between the F-met and the glycine, so now we have a dipeptide. We have the first 2 amino acids. All right. Then the ribosome translocates or moves to the next site. And again, this is going to involve GTP and its hydrolysis to GDP and an elongation factor G. Now, what's happening here is the initiator tRNA is now going to get kicked out of the exit site because it's lost its amino acid. All right, because this dipeptide is now associated with the tRNA that came in there previously. It's now sitting at the P site. So the next tRNA can come in at the A site, transfer it on to the glycine, translocates, and the process just repeats over and over again. Now, the interesting thing, so ribosomes have all these proteins and and they also each have 2 ribosomal RNAs associated with each subunit. So there's 4 ribosomal RNAs. People working on this problem couldn't find the protein on the ribosome that catalyzes this very important reaction. The peptidyl transferase is the enzyme that would make the peptide bond. Probably one of the most critical steps in this whole process. And they kept purifying the proteins and they couldn't find it. Turns out it's catalyzed by the large ribosomal RNA. So this is another example of a ribozyme. So this is an RNA in the subunits that actually has catalytic activity and catalyzes. It has the peptidyl transferase activity, and it's what catalyzes the peptide bond formation. And that process just continues until the ribosome encounters one of those 3 stop codons. So at that point, and in E. coli, they just have 1 release factor. In other words, there's 1 protein in E. coli for each of the 3 stop codons. So when the ribosome encounters the stop codon, there are no tRNAs. It is these release factors that now enter in through the A site, and they promote the cleavage. In other words, basically, they're going to let the polypeptide be released from the tRNA. The 2 subunits have to be released. So everything dissociates, and then the whole process can recycle again. Okay. All right. Some key differences here. If we just want to compare between eukaryotic and prokaryotic cells. So again, in bacteria, initiation is through this formulated or F-met methionine, formulated methionine or F-met. This is not in eukaryotic cells. The other big difference is, as we talked about numerous times, in prokaryotes, transcription and translation are occurring in the same immediate area. They aren't sequestered. So this has important implications for gene regulation. So this will be something we'll talk about at the end of chapter 16. There's a mechanism of regulation called attenuation that actually takes advantage of the fact that transcription and translation are essentially coupled in prokaryotes. But of course you can't do that in eukaryotes because the processes occur in different places in the cell. The other is mRNA tends to be very short-lived in the bacteria. You basically transcribe it, translate it, and degrade it. That is not true with eukaryotes. Some eukaryotic mRNAs may be degraded within a couple of minutes. Others are stable for hours. Some are stable for days. So it really just depends on the particular system you're looking at. The other is, while the overall process is very similar, you have the ribosomes, the large subunits, the small subunits. The actual proteins themselves are very different between prokaryotes and eukaryotes. So this has really been the basis of antibiotic development, is to exploit the differences, the biochemical differences between the proteins in prokaryotes, the bacteria we're trying to kill off, and the corresponding proteins in our cells. Okay. The other thing is something called polyribosomes. All right. So the ribosome is associated with the mRNA, but as soon as one ribosome clears the start codon area, another ribosome can join on. So you can have multiple ribosomes translating the same mRNA. So it's an amplification step. You can get many, many copies of the protein from one copy of the messenger RNA. So polyribosomes are simply an mRNA molecule that has 2 or more ribosomes associated with it. The more ribosomes associated, would that be indicating a more efficient or less efficient mRNA? It would be more efficient. So the larger the polyribosomes, which actually reminds me of another story. Okay, here's how you lose your job. So, as I said before, I did my master's degree with a couple of scientists who actually did the work on, a lot of the main work on the cap structure. So they did a lot of work on translational control and looking at polyribosomes. And we would separate them in a centrifuge gradient and run them through a spectrophotometer. And this was back, again, old days, so we had chart recorders. So you tended to get these little things that look like fingers, like you traced around your hand. All right. Well, in one of the labs that Dr. Weber was working in when he was up at Connecticut, they had a technician who just couldn't get this technique to work. And so one time in one experiment, just out of desperation, she just took the baseline adjustment and drew what she thought it would look like. Then she joined the ranks of the unemployed. Probably never worked in another lab again. So the best way, if you're working in one of these labs, if you want to just destroy your career, just start fabricating data. Related to that, but okay, if you want to see some of the pressure people are under, the NIH has a division that investigates scientific misconduct, and you'd be surprised at how many people have submitted fraudulent data in grants and publications because tenure's coming up, or they need that grant, or they're going to lose their job, sort of thing. And under extreme conditions, people do extreme things. But it is the single best way to destroy your career in science. All right, so let's take the same picture. This is actually an iClicker question. Okay. All right, this should only take a minute. All right. And the answer is why? Because you just took a wild ass guess at it. How can you tell this is from a prokaryotic cell? Oh, I think I just saw a couple of light bulbs going off here. What's happening? Is transcription complete yet? No, transcription. So this is what you see in bacterial cells is even before the messenger RNA is completely synthesized, the ribosomes are already beginning to translate it. Okay? All right. Here's a very important point. It actually ties into biotechnology and the synthesis of recombinant proteins. Today, in Dr. Weissbach's career, he's probably purified, I don't know, hundreds of proteins. Many of it being the old standard methods where you use column chromatography. They still do this in biochemistry, the ion exchange columns and the gel filtration columns. And okay, well, that all works fine if you're going to isolate a protein that's fairly abundant in cells, and you have to have an enzyme assay to be able to purify it. But in many cases of so-called reverse genetics that we do today, we find the protein of interest, but we have no idea what it does. So the easiest way to make this is to make a recombinant protein and make some cell, usually like E. coli, make the protein for you. So for instance, we do this in the biotech lab. We make E. coli pump out a Drosophila protein for us. But the important point is, a lot of proteins, and especially in eukaryotes, aren't active as they come off of the ribosomes. They have to have post-translational modifications. There may be some clipping with some amino acids removed, some phosphorylation, glycosylation, which is actually the addition of carbohydrate groups. So especially like cell surface receptor proteins tend to be heavily glycosylated. And so until these chemical modifications are done, the enzyme is inactive. So why might making these proteins in E. coli be a problem? Can E. coli carry out all those reactions? Most of them no. So if your protein doesn't need these kind of reactions, E. coli works pretty well. If it is going to have these kind of modifications, then you want to use a eukaryotic system. For instance, there's a insect cell system that uses a virus called baculovirus, and you can use that to make recombinant proteins, but since it's a eukaryotic cell, it can carry out these types of modifications. The other important point is one of the reasons that proteins can't renature themselves after they've been denatured is they're taking on this three-dimensional structure, which is really taking them to their lowest thermodynamic state, their most stable state. But they're beginning to fold as they come off the end of the polypeptide. So that folding is not being influenced by the amino acids that have not yet been added. Once the whole protein is there and the whole thing is taken apart, then it just sort of randomly collapses on itself. The other was the discovery of molecular chaperones, probably about 10, 15 years ago. Turns out these are mainly the heat shock proteins. So it took a couple of decades to figure out what these heat shock proteins were doing. They are molecular chaperones. So what that means is, under normal conditions, these are proteins that now associate with the nascent polypeptide as it's coming off the ribosome and help it to fold into its proper structure. Then under stress conditions, these proteins, these molecular chaperones, interact with key proteins to help try to keep them from denaturing during that stress, like thermal stress, something like that. So there's these extra proteins that are needed for a lot of, in many cases, to get the right conformation on the proteins. So a couple comments about antibiotics. So as I said, the proteins in the ribosomes, in this whole translational process, are quite different biochemically in prokaryotes than they are in eukaryotes. So this has been taken advantage of in the development of antibiotics. So there's a range of antibiotics that all work to, here's just 4 examples of antibiotics that work to block translation in bacteria, but they're specific to the proteins in bacteria. So for instance, tetracycline blocks that entry of the tRNA into the A site. Chloramphenicol binds to the large subunit of the ribosome and prevents the peptide bond formation. Streptomycin binds to the small subunit and blocks initiation. And erythromycin blocks the actual translocation or the movement of the ribosomes along the mRNA. Okay. So since these drugs are targeted to bacterial proteins, why do you sometimes have side effects when physicians, let's say, prescribe these for their patient? Especially tetracycline can have some. Should these drugs have any effect on your cells? Exactly. Remember, the mitochondria are more prokaryotic-like than eukaryotic-like. So especially at higher dosages, these can interfere with some mitochondrial functions. And some people are going to be more sensitive to it than others. All right. Let's try this question. Mm-hmm. Okay, it looks like 1 minute should be good for this one. Okay. So what's the answer? Okay. How do we do on that one? Okay. Most of you got it right. All right. So it's moving rather quickly through that chapter, mainly because the next 2 chapters are on gene regulation. And this is where things get a little trickier, a little more involved. They're divided up. So chapter 16 is going to be on prokaryotic gene regulation. 17 will be on eukaryotic gene regulation. So any questions on translation? Okay. Not enough time to formulate a question. All right, you can think about that. So again, what I want to do is I'm going to take the next 15 minutes. I want to introduce chapter 16 and do the easy stuff so I can save the hard stuff for Wednesday. Not hard stuff. I know what it is. It's not easy. How many of you already know everything about the lac operon? I could ask you any question about the lac operon. How many of you even heard of it? You've heard of it. How many of you just know everything about it? You know exactly how it works? I could ask you the question. That's a pretty small number. Okay. So, and since it tends to come up in other classes, by the time we get done on Wednesday, I'm going to tell you everything you need to know about the lac operon, and that's going to help you on exams in other classes too. Okay? So this is chapter 16. So we're talking about gene expression in prokaryotes. It's going to be very different in prokaryotes than it is in eukaryotes. Okay? So That's why it's better that it's actually divided into the two separate chapters. So this is just in bacterial systems. And again, we're going to use E. coli as our example. Having said that, one area of similarity between prokaryotes and eukaryotes are DNA binding proteins. What you're going to see over and over again is this story of a factor, a transcription factor, a transcriptional activator, a protein that's going to bind to a specific DNA sequence, and then there's some outcome from that. So these DNA binding proteins are common in both prokaryotes and eukaryotes. That's a common theme, but they're used in different ways. So the first part of this is just to look a little bit at the characteristics of these DNA binding proteins. Okay. So we got to think about protein domains or motifs. This is not the whole protein. This is just a sub-part of the protein. Okay, so it's like this is just the master bedroom. It's not the whole house. It's just a part of it, but it's a part that's going to help tell us what the functionality of that protein is. Okay, so these DNA binding proteins, as it says, they're on the order of maybe 60 to 90 amino acids, and at least some of them will actually make direct contact with the DNA. So these motifs are larger structures. So let's see if I can complicate this. The alpha helix and the beta pleated sheets that we talked about are used as part of to make up part of these domains or motifs. And they make up part of the whole protein. So what I'm going to do is show you now a couple of the most common DNA binding motifs. And what you'll see is that alpha helix especially is a common theme throughout this. So maybe it'll make a little more sense when you see the figures. So the three I'm going to show you is one is called the helix loop helix, zinc fingers, and leucine zippers. These are three of the most common. So what these are motifs, if you see this, if you're looking at the structure of a protein, you don't know what it does. If you see that it has one of these or two or more of these domains, then you get a good sense that it's probably some kind of a DNA binding protein. So the first one is a helix turn helix. This is just two alpha helices. So this is secondary structure. Alpha helix we talked about. We have two helices connected by some length of polypeptide. The key here is these helices fit into the grooves on the DNA. So you have one in the groove here, one over here, and then you've got some amino acid sequences that connect the two together, and they're going to differ from protein to protein. Again, this isn't the whole protein. This is just a structure that you're going to find. But if you see this on your particular protein, it'll suggest to you that it's a DNA binding protein. Zinc fingers were actually the first ones to be discovered. And they got their names because one is the motif, when you look at it in 3 dimensions, looks like fingers, and it has a zinc atom coordinated with it. So it got the name zinc fingers. These fingers are just the right molecular size and structure to fit into the grooves and reach down and read the bases along the base of those grooves, especially the major groove. Okay, so again, you see more of these helices that fit in, and now you're putting more together, making it a little more complicated. But if you see a zinc finger motif, it's going to tell you this is probably a DNA binding protein. And the third one that you see a lot of times on transcription factors are these leucine zippers. So again, it's an alpha helix, but it has leucines positioned in just the right places, so literally 2 of these can come together and literally sort of zipper the 2 proteins together. What you see with the many of these DNA binding proteins act as dimers. They act as a pair of them. And so the leucine zippers allow these 2 copies of this polypeptide to interact with each other and basically tell each other they're there. There's other proteins, for instance, in the cell membranes that are inactive when they're separated, but if they come together, then they form an active complex. So that's where this leucine zipper is a common motif for proteins that facilitate protein-protein interactions. Okay. So as a prelude to what I want to talk about in terms of real gene regulation on Wednesday, we have to talk about operons. Operons are a gene structure in bacteria that you do not see in eukaryotic cells. It's a very straightforward idea. Cluster together all of the genes that you need for a particular process and put them under control of one switch. So even histidine, which I think has 10 or 11 different enzymes, you take the genes for all the structural genes that code for all of those enzymes, put them all together, and put them under control of one promoter. So it would be like controlling the lights in this building. Put all of the lights on one switch. It's not particularly efficient, but if I need lights in this room, I can turn on all the lights in the building with the flip of one switch. All right. You don't see this type of gene organization in eukaryotes, so it's going to be unique to bacteria. All right. Okay. Here to just sort of introduce you into some of the jargon. So because what we're going to have to deal with on Wednesday is things like repressible, inducible, repressible, all this sort of different ways of gene regulation. The key is there's going to be a regulatory protein and there's going to be a DNA binding site. All right. The regulator gene that codes for this protein is not part of the operon. It can be anywhere else. It's a diffusible entity. So that gene can be encoded anywhere else that is not part of the operon. But the product of that gene is needed to control expression of that gene. So we're going to talk about the lac operon in some detail next time. The cell needs the products of 3 structural genes, LAC-A, LAC-Z, and LAC-Y, to be able to metabolize the lactose. But without the lac repressor, it can't regulate expression of it. So that's part of the regulatory system. So you transcribe this gene, you translate it, and you get the regulator protein. The operator is a DNA sequence. It's the target for that regulatory protein. So the regulator protein is going to interact with the operator sequence. The operator is an integral part of the operon. So to show you this in a very generic sense, and then next time we'll be starting to tease this apart, here's a typical bacterial operon. So very similar to what we've seen before. Here are the structural genes, A, B, and C. The transcriptional start site would be right here. And so these 3 genes are transcribed. This is an important difference too. In prokaryotes, in a system like this, there's one mRNA that's going to transcribe all 3 genes. So all 3 polypeptides are going to be encoded in that single messenger RNA. You do not see this in eukaryotic cells. In eukaryotic cells, each mRNA codes for 1 polypeptide. So in this operon, when the system is active, you get transcription, 1 mRNA that codes for all 3 proteins, and then down here you get translation, you make the 3 proteins, and they function in the biochemical pathway. Located between the promoter, we talked about the promoter is immediately upstream, between the promoter and where transcription begins is the operator. This is where the regulatory protein is going to bind. All right. In this particular case, this protein, and what we'll talk about, this protein can be in an active or inactive form, so it can bind normally or not bind, or other molecules can influence it. But for instance, if this protein binds to the operator and it acts in a negative sense, it basically becomes a roadblock, prevents transcription and shuts the system down, and it won't be turned on until the protein is taken off. So it's going to be this regulation between this regulatory protein and how it interacts with the operator. Okay? There's going to be 4 different ways of doing this. Negative inducible, negative repressible, positive inducible, positive repressible. I'll end with this comment so you can think about this. This is where we'll start on Wednesday. Negative mean, instead of going, oh my God, what's this all look at? Break it down. Negative means that when the protein is bound to the DNA, it will block transcription. Positive means when the protein is bound to the DNA, it will enhance transcription. Okay? Inducible means we're going to have to do something to make transcription happen, and repressible means we have to do something to make it stop. And that's all we have to do with it. So we'll leave it at this point. So we'll finish up chapter 16 and should still have plenty of time to answer questions about the exam. So we'll see you on Wednesday.